Hello, everyone, and welcome to this week's lab investigation. My name is Shannon Reeves, and this is my email below, and this is Ravioli. We're here to take you through this week's lab investigation about anaerobic respiration. So, before we get started, let's go ahead and go through some announcements. This is this week's investigation question. So, why do yeast cells metabolize some sugars at different rates than others? And we will go into the specifics of that lab. But you'll see a lot of these same announcements on the photosynthesis lab next week, so don't be surprised if you see them again, but they're good reminders, so it's okay if you see them. But word limit. Oh, let me move my view. Okay, so word limit is set at 350 from here on out. So no exceeding without losing points. You all have had some practice in being able to do that, but following the rubric will be crucial for this. So keep in mind that the lab summary is not a lab report. For those of you that have had chemistry or other sciences, you've probably gotten pretty used to the detail that's required of you in lab reports. This is more like the abstract of your lab report, where it gives you a broad overview of each of the sections. So it's a summary, it's not detailed. So don't get caught up in a lot of the minutia. That's where a lot of your word count gets rough. So don't get into details that aren't mentioned in the rubric. Grades are in, so remember to look at your comments in your lab summary submissions. I've been doing quite a few of them, but by now you probably know that your macromolecules lab is the only one that I'm going to be leaving comments for. This is just because I am finding it difficult to get through all the lab reports in a timely manner while leaving comments that help you guys. So I will still be following the rubric and giving you an indication of what you lost points for. I just won't be leaving thorough feedback on how to make that better. Hopefully by now you have a better idea of what that should be, so it shouldn't be a big deal, but you're always welcome to come into office hours and talk to me about your lab summary if you do have questions. Just keep in mind that I won't answer emails about that because that's simply doing the exact same thing. I am just writing out the commentary that would be there otherwise in the lab report. So that's what takes so long. So I won't be doing that. But if you are still losing points on your lab summary, it's probably because you're not addressing points in the rubric, and those add up pretty quick. So I highly suggest you have the rubric out while you're writing so that you're making sure to address each component of the lab summary that we want you to hit. Also, just as a major announcement, my cat does not eat garbage. I was not very clear about that this last week. Ravioli eats junk food, which I happen to call it garbage food. She doesn't actually eat out of the garbage can, so that's good. She's healthy that way. She just happens to eat kitty kibble more than she likes to eat protein that we like to give her. So I just wanted to clarify so I don't seem like I'm a terrible pet owner. Also, this picture is pretty cute. Okay, reaching out for help again. I will try and move this. So please do not wait until Friday to reach out for help. I have very little time on Fridays just because my Fridays are whenever meetings usually happen. So from about 8 a.m. until 4:30 p.m., I have meetings straight through. I don't even have a lunch hour. So I won't be answering any emails on Fridays. But this is a good reminder that you guys are in college. You have a whole week to do these assignments. So you have plenty of time to get a hold of me as long as you don't procrastinate on your assignments. So let's go through some of the protocol for our lab experiment today. So you guys will be using a CO2 probe for this experiment. We'll be using it next week for the photosynthesis lab. So you'll see the same picture that reminds you that you need to warm up your lab probe and set it on low. So this is just a reminder not to let the equipment tip over because the probes are pretty fragile, but this is the picture of the chamber that you'll be putting the yeast in order to do your experiments along with the sugar. So we put the yeast along with the sugar inside of this chamber, and Logger Pro again will be helping us to identify the CO2 parts per million that is present in the actual chamber. So after 2 minutes, the CO2 reading is usually around the range from 300 to 800. Once you get those values, you're going to be comparing those values for each sugar. So this is an example of what Logger Pro will look like with the data. Again, we're going to be giving you data, but this is the same with your photosynthesis lab. You need to take out the first 5 minutes of the trial. So as you can see here, this person has highlighted the last 10 minutes of the data. This won't look quite like your data. You'll be given different data, but the minutes will be indicated. So just be careful that you don't use all of your data in there, because Logger Pro takes some time to set up, and so does the CO2 in order to get yeast respiring for a while. So we'll be taking the last 10 minutes of our data in order to find the rate of CO2 production. So just as a reminder, a rate is over time. So your time right here is 10 minutes. All right. Sorry, I have a little bit of a scratchy throat today. So when you're finished, this is a reminder to go ahead and clean up. You rinse out your respiration bottle and everything as needed. But this is essentially what it will look like to mix all of our trials. So we're going to have each different combinations of sugar with your yeast. So our very first combination will be our controls. So our first set of yeast will be water that will be added in addition to the yeast. So we'll have positive and negative. If we're to have yeast that's respiring anaerobically, then we won't have sugar. So, or I'm sorry, if we don't have yeast that is respiring anaerobically, it will not have sugar. So this is our negative control. This is going to be our example of something that won't do a really good job of respiring, because we need sugar of some sort in order to do so. Glucose stands as a pretty good poster child for good cellular respiration. We know glucose is a really common sugar, and it works in yeast just like it does in humans. So glucose will be serving as a monosaccharide. Our disaccharide will be sucrose, which is a glucose molecule, which is bonded to a fructose molecule. So you will want to identify which one of these positive controls you'll be using, and we'll talk a little bit further about that in future slides. So lactose is another disaccharide. So it is made from glucose and a different sugar called galactose. You have the same bond that's bonding these two molecules together. That's your covalent bond. So you can compare your three controls here to another disaccharide, or you can compare your three controls right here to a monosaccharide, raw sucrose. So the idea is you could compare your three controls by saying that you want to know the difference between disaccharides and how they might respire. Given the disaccharides, you'll still want to include your water as your negative control and your glucose. Or you could do those three versus raw sucrose. So you may just see as a monosaccharide, does raw sucrose, because it's raw instead of processed, does it have some sort of minerals or vitamins or anything additional in there that might aid or inhibit respiration? So that is where your hypothesis will come from. Just keep in mind, in order to use any of these sugars, that just like humans, yeast have to be able to digest them, and digestion is aided in an enzyme. So you have to be able to have the enzyme in order to digest any of these and use them in respiration. So choose between one of these two sugars. You won't use both. You'll use all three control groups. Again, just to be clear, three control groups. You'll have to identify which ones are negative and positive, and then compare it and have a good causal statement in your hypothesis about why you think it's going to go one way or the other, and why you might be interested in lactose over sucrose or sucrose over lactose. So you're going to have to do a research proposal part of your background. So before you actually write your summary in the same Word document, but maybe on the first page or on just right before the summary, go ahead and identify the following. You'll want to write out your hypothesis, your control groups, your experimental groups, your independent variable, and your dependent variable. So be sure to do these things both before as well as in addition to including them in your summary. Because you identified your hypothesis in the beginning, this doesn't mean that it doesn't need to go in your introduction. You need to include all of these at the beginning, almost as just bullet points that let me know that you know the parts of your design, and then go ahead and include them in your summary as well. So when your research proposal has been completed, you're going to select the data that you want in your actual experiment and complete the lab summary. But first, I wanted to go a little bit over the methods section. So we've been talking about how to write different things in our lab reports. And last week we covered the introduction. So this week I would like to cover the methods section. If you remember in our experiment, we were talking about different areas of the house that Ravioli likes to sleep in and comparing them. So we're going to continue doing that. This was our introduction. Just as a reminder, Ravioli enjoys sleeping almost as much as eating. It's one of her favorite hobbies. We've noticed there's specific spots around our house that she favors. In the past, sunshine and warmth have been a common factor in spots that Ravioli picks. We are curious if warmer temperatures are what attracts Ravioli to her favorite snuggle spots. We hypothesize that Ravioli's sleep spots have higher temperatures throughout the day than other areas of the house. This experiment was carried out by taking the air temperature in the areas Ravioli was taking naps and comparing those temperatures to other spots in the house. Ravioli was not pleased or cooperative during the process. So the very first part of the methods section, again, I'll move this slightly. There's no good spot to put me, so I'll put it down here. The methods section describes the groups being studied. So this means identifying your experimental and control groups. A lot of people have been writing in their summaries, we had a control group and we had an experimental group. This isn't enough. You actually have to identify what they are. So in our experiment, I explained them. In order to test the relationship between temperature and favorite nap spots, we took the temperature of each napping spot as well as the recorded time that Ravioli spent sleeping in each spot. So just as a reminder, that first sentence right there identifies your independent and dependent variables right off the bat. So if you want to introduce your method section by doing a similar sentence, you can. But if you want to just jump in, or if word count is really a big factor, then you may just start right on the second sentence that I have there. Ravioli's favorite spot in her cat bed next to the piano serves as our control group. The three remaining spots, the windowsill, her scratching post, and her orange chair, served as the experimental groups. So next, next, describe the protocol being used. So these are the steps to your experiment. Don't give too many details though. Word count is precious. So think about things that are specific to this experiment. Honestly, this one, it was hard for me to come up with very much because it's pretty self-explanatory. But I put to compare each napping spot, the 3 napping spots were cleared of any obstructions and left open for Ravioli. Participants watched the napping spots for opportunities to catch Ravioli sleeping. In our next part, we describe the type of data that's collected. This is pretty simple. Temperature of each napping spot with an infrared temperature radar when Ravioli was found sleeping there. That is not a complete sentence, but I am like 15 minutes into this, so I'm going to move forward. The temperature of each napping spot was taken, there we go, with an infrared temperature radar when Ravioli was found sleeping there. I must have edited that and did not notice that I didn't fix it. So this is just a simple identification of what actual data you are taking. This may not be the data that you include in your graph, like in this lab. but it's the way that the reader can know how you found that value that you're going to put on your graph. So lastly, how was the data analyzed and displayed? This is two things. This is the most common thing that students leave out of their lab summaries. This is how you prepared your data, meaning analyzed it, and it also tells your reader how you constructed your graph. So in my experiment, raw temperature was what we were looking for. So I just said because temperature fluctuates throughout the day, during Ravioli's naps, the beginning and ending temperatures were taken and then averaged. That is now the calculation that we used to analyze and put this stuff in our graph. So analyzing it may be as simple as averaging it. Average temperature of napping spots was compared in a bar graph. That just simply tells our reader how we constructed our graph. So in the end, we have all four parts of the method taken care of. I prematurely went on to the next slide, but focus on what I'm saying, don't read. So this gives us a good idea of how we did everything. So the point of the method section essentially is if the reader doesn't have the experimenter with them or the person that created this or a TA or a teacher, then can they recreate this experiment sufficiently on their own just from the summary? So now I am your reader, so you don't have to include details about cellular respiration that maybe somebody else wouldn't, or aerobic respiration that somebody else may need. But in our methods section, keep in mind that certain things like we talked about how we prepared our data. You won't be giving me the amount of CO2 that was produced. I don't want the overall amount. I don't want the final amount. I want the rate. So this will be taking the first minus the initial over time. So the difference, how much it changed over time. That's your rate of CO2 production. I just gave you your calculation. I won't be as generous in future things, but be sure to include that calculation in how you analyzed your data. So one more time, we're going to give you a paraphrasing practice. So, oh, okay, you can see that. That says plays, not praise. Here is how we want you to summarize this into one sentence again. So keep in mind that you want to take the most relevant information. Last time, whenever we were talking about the enzyme stuff, I got a lot of students that told me stuff about supermarkets and how they're interested in keeping their fruit fresh. That was probably information that would have been good had we had more time to talk about this stuff. But you want to take the most relevant information to this summary right now. Paraphrase that from this stuff and then put that in your assignment. So usually this is going to be in your introduction or in your conclusion. If it's in your introduction, it's going to help support your hypothesis or tell the reader a little bit about what background information they need to know in order to be somewhat interested in this topic. Or it's put into your conclusion as supporting evidence for what you found out from the lab. So keep in mind that we have how to cite and how to write a lab summary that are available under your lab resources module to review before or during writing. And I highly, highly suggest that you do this. Here is what you need to include in your literature cited. Again, this right here is not complete. You'll have to do how to cite, or you'll have to look at the how to cite PDF that we've given you and put this information in order. So this week you're going to design an experiment, complete your research proposal. So this just simply is that 5 bullet points beforehand. You don't have to complete it separately, include it in the same document. You'll carry out your experimental design and collect data, analyze it, and put it into a graph. And then write your summary, make your Flipgrid video, and upload it to Canvas. So just as a reminder, it should be uploaded by 5 p.m. And your Flipgrid video should address some of the following things. So a brief summary of your experiment, something new that you learned as a result of this activity, and how the concepts in this lab are related to what you're learning in lecture. I have additional prompts that you can address if you are wanting to address some of those that are available on Flipgrid. And that is it. So if you have any questions, again, I'm available through email. We can meet in person or over Zoom. And otherwise, I hope you guys have a great week.